Now that we've introduced some ideas about why population genetics, the fourth big area of genetics, might be important or interesting, it's time to think about some of the theory. And also, we're going to use that theory, you're going to need to be able to calculate allele frequencies. We're going to talk a little bit about that. So first, a couple of definitions. For our purposes in population genetics, because the genetics is the big word there, we need to have individuals that are capable of breeding. So unlike maybe an ecological population, which could just be any census of number of organisms, plants, or animals that are out there, in genetics, we need to have those that can either interbreed or potentially interbreed. So they need to be able to share genetic information. The reason for this is that we're interested in studying what's known as a gene pool. And the gene pool is the total amount of genetic variation, usually in terms of alleles that are possessed by the reproductive members of a population. So what we're interested in are things that can be passed on. So what's contained at this point that has the potential to be passed on as genetic information. So that's what we call our gene pool. Again, we used to think of gene pools in the simplest sense in just presence or absence of certain alleles or the frequency of those alleles, but it can mean all the information. So put this together, and I told you I would give you another definition. We had that original one, a very common definition. This one's a little shorter, but we'll talk about the idea. It's the same basic idea. It's a branch of genetics, right? Involved in the study of the mechanisms responsible for modifying gene pools, which is essentially exactly what the last one said. It just doesn't specify here what those mechanisms are. But we're looking at modifying gene pools. Gene pools are never static. The amount of genetic information, the way it's distributed among a population, a group of individual organisms, is never the same generation after generation. So we want to know what makes those changes occur. And this is schematic I'll use multiple times, but it's sort of my way of representing what's going on. We have a gene pool, and we have some gene pool at another time. And to get from this gene pool to this one, you have to have reproduction, and we're considering sexual reproduction as we've been throughout the entire course. But this is going to filter through what is known as population size. It's going to play a big role in determining which one of these other mechanisms can play the largest role. And drift, if the population size is very, very small, drift or chance will be very important. Mutation may or may not be important, and that may depend on time and as well as size. Selection then and drift kind of work together in various ways, and it depends again on directly on the population size. So these are all fit together. Now it's nice to have a little schematic like this, but obviously you need some kind of theory for this to work, right? If we want to be able to test the amount of selection that's going on or the effect of drift or whatever, we need to have a theory that we can make predictions from to do those tests. That's how we do science. And we have a theory. It's been a theory around for over 100 years, and it's a really important idea, okay? So let's go back to 1900. We've got the rediscovery, using air quotes here, of Mendel. Number of individuals, these are three prominent ones, De Beers, Corinnes, and Tishmark, have been working on basically doing very similar types of studies and working with plants. And when they did this and they got to digging around through the literature, as we often do, they go, oh, well, my goodness, that was published almost 40 years ago and by Mendel. So they rediscovered Mendel's work, brought it forward, and verified that it was correct. Okay. Now this started a flurry because now we had, by this time, 1900, we knew where there was chromatin. We still didn't know what DNA, how it really worked or anything, but we knew there was chromatin. We knew that there was things going on that we could understand about genetic systems. We could do crosses. We had model systems coming about. And so people were really very interested in Mendelian systems. Now you may remember just from a simple Mendelian setup, if you have a dominant and a recessive, you'll get all dominants. You cross those dominant hets together, you should get these 3-to-1 ratios. So people started running around looking for 3-to-1 ratios, and sometimes they would find it. Certainly if they set up the right cross, they would always find it. But other times when they just go out in nature looking, they wouldn't find it. And so it became a real question, is Mendel correct? And there was some ideas that maybe these, Mendel had just stumbled on a few traits that had this outcome, that many didn't have it. And it was really a question. In fact, it still not too long ago carries through. Back when the internet was relatively new, I know you think it's been around forever, but it wasn't. And when I was first here at Auburn and the internet was becoming common, I wanted to learn HTML, so I wrote a little website that said "Ask a Geneticist," and it was just a simple little website you filled in, and it would email me a question with your return, and I would try to return answers, and got a lot of interesting questions. It was geared to K through 12, so it was always kind of fun. But one day I got this very nice email from this teacher, and what she had done is she had picked out some human traits, such as attached earlobe, flexibility of fingers. I can't show this one, but a dimpled chin. Surely can't show this one. A widow's peak. Some of these others that known to be simple dominant recessive. And she'd gone around her class and had her class collect data, and it didn't. None of them. I think she had 6 or 7 traits showed up to have a 3-to-1 ratio. So she sent the students out to the whole class, the whole school, and they collected quite a bit of data. Still couldn't get a 3-to-1. And apparently this was a progressive school at the time, and they had a webmaster. So they set up a little program, and they had schools from all over the world do the same little project and send in the data. And at the end of all this, they said, I still rarely found 3-to-1 ratios. And so by this time she emails me and said, what's going on? I mean, we've heard, for 100 years that we should be seeing these 3-to-1 ratios. And I said, well, yeah, actually that problem was solved in about 1908, and it's a very important problem. And it relates also to this same question. If you get 3-to-1s under Mendelian process, how do we persist, particularly if these are really like deleterious alleles? What if they're recessive lethals? Why are they even there? So it's the same relative question. Well, the person that had been working on this problem, had been forced to work on it, was Reginald Punnett. And Punnett had been working in the late 1800s on crosses, and then by the 1900s, with Mendel's rediscovery, he was a Mendelian. So remember these big conflict that went on between the biometricians and the Mendelians. And he was working up here before Mendel was rediscovered. Once Mendel was rediscovered, he and Bateson had actually worked out this idea of linkage. Now, they didn't call it linkage at the time, but that's what they were working on. Now, you may recognize Punnett for being the famous Punnett square, right? That we've used when we did transmission genetics, same person. Well, in 1908, Punnett was giving a talk at a meeting, and people were just really asking, wait, why don't we see 3-to-1s? Why not these recessives, these deleterious things, why do they persist? Why are they there? And he had been thinking about this problem for quite a while, but he couldn't get an answer to it. And so he approached a good friend of his named Hardy. And Hardy was one of the premier mathematicians in the UK. I mean, he was a pure mathematician and was just an outstanding mathematician. Pure mathematicians are people who want to do math, but they don't want it to be applicable to anything. That's what a pure mathematician is in many cases. But Hardy said, well, I don't know, let me think about it. So he sat down one weekend. He said, okay, well, here's the first issue. Sure, you can get 3-to-1 ratios, right? Just like we do in our Mendel's Big Six, but think about a population. That's not the only cross that's going on in the population. Sometimes you've got this one, this one, this one. You've got all 6 of these common crosses that could be simultaneously going on in a population. So you can't think about the outcome from one of these results. You have to think about them all. So he set up a very elegant little chart, and we're not going to worry you with it here. If you take population genetics, we'll actually derive it. But he set up a nice little chart showing the outcome of each of these crosses, set up the probabilities for those. And when he did, he came up and realized that there was a very common outcome. And that became known as the Hardy-Weinberg principle. It's Weinberg because a German physician had basically figured it out at exactly the same time and published on it at the same time. So Hardy and this German physician are credited with this idea. So what did he say? He said, well, okay, all this is going on simultaneously. We can't be thinking about single ratios anymore. What we need to start thinking about are what we're going to call allele frequencies. We need to think about the frequency. How often does one allele occur relative to the other? And if we've got a multi-allele system, we just expand it out. But he was thinking in terms of a 2-allele system to start with, right? And he sat down and said, okay, it's a very basic, simple math here. If you've got 2 things, okay, the proportions of those add up to 1. If you've got 20% of A, you better have 80% of B if they're going to add up, right? And so he just set up a simple equation, said P plus Q, use P and Q equal 1. he said, okay, there's something important we can do to that. We can do a binomial expansion, and those of you very familiar with that, of course, you expand one, you square it, you get one. You expand p plus q, you end up with this equation: p squared plus 2pq plus q squared equal 1. Very important equation in geometry and other places, but here it's going to play an important role for us. This literally became the Hardy-Weinberg equation. And there are simple extensions that you can do if you've got 3 alleles or 4 alleles, but it's simple form. Now, what does this mean? What do these relate to genetics? Well, p squared is the frequency of the homozygotes in the population, one homozygote. Q squared turns out to be the frequency of the other homozygote. Traditionally, this p squared has been the recessive. Used as the recessive. It doesn't really matter, but traditionally in working problems, you'll just use q squared as your recessives. And then, of course, 2 times p times q is the frequency of the homozygote, the heterozygotes in the population. Frequency of the heterozygotes. So you've got the 2 homozygotes covered, and you've got the number of heterozygotes. And with 2 alleles, you're only going to have 1 heterozygous type, right? Big A, little a. 3 alleles, you might have more, but you'll have 2 PR, and it doesn't matter for us, okay? All right. So if we took some words, I know math is tough. We take some words out of that. What does it mean? Okay, it says at a single locus, so we're talking about just the alleles, one locus. After one generation of random mating, you can represent the genotypes as a simple function of the allele frequencies. And that function is nothing more than a binomial, or if you got multiple alleles, a multinomial function. It's that p squared plus 2pq plus q squared. So all you need to know are the allele frequencies, and you can figure out the genotypic frequencies, or vice versa. If you know the genotypic frequencies, you can figure out the allele frequencies. Now, this random mating part becomes very important up here, okay, as it was developed. It predicts, the Hardy-Weinberg predicts that there will be a genetic equilibrium that's reached after one generation of random mating. What that means is you can take extreme cases, and we do this in courses sometimes. You can take a population of fruit flies. You can take two alleles. So this one's true breeding for short wing, and this one's true breeding for regular wing. And let's just pretend they're codominant or something, so you can tell everybody. All right, so you got 50 flies over here that are normal winged, and 50 flies that are short wing, right? Put them in there. The first generation, you got no heterozygote. I mean, without breeding, you got no heterozygotes, right? So the frequency of the allele of q squared is equal to this, 0.5 and 0.5. If they randomly mate, you open the jars up, connect a way for them to mate, and you let them mate for a generation. After one generation, you should, in that next group, the next gene pool, the next group of progeny, you should have about 25% of them being normal, about 25% of them being the small wing, the reduced wing, and then 50% being heterozygous, whatever that may be, okay? And if you let them randomly mate, they will continue forever at that particular form. That's what the Hardy-Weinberg says, okay? It's this genetic equilibrium. And it's a really important idea, because what it means is two things. One, natural systems, if you have random mating or even close to random mating, they move toward wherever the frequencies will push them, and they stay there. It takes a lot of effort to move them away from that equilibrium. It's a very strong equilibrium point. So it has to take a lot to move them, right? So selection or drift or whatever force is working needs to be very powerful to make it any real effect over time. Or it needs to be gradual and slow, which is some of the ways that selection works, and it will still push it. Okay, but it keeps trying to come back to this equilibrium. So push it over here, it tries to work its way back to the equilibrium. Okay. The other thing it means is that if I go out and I look in nature and I find out that I've got a set of genotypes that are not in Hardy-Weinberg's equilibrium, right? And we'll work on the next lecture on how we actually test that. It means that something really powerful and interesting has happened to that population, at least as a gene pool of that population. So it's a really useful tool for that. Now, just to point out a term so that you're aware of it, random mating here is truly panmitic mating. Okay, panmitic mating means, in the sense of random here, that the genotypes, these Mendel's Big Six, those crosses, will occur at the frequency related to the alleles. Okay, so if you got an extremely rare allele, okay, it's really, really rare. It's like 1%, is all, right? It should only be involved in 1% of the matings, wherever type of mating it was over here. It should only be involved in 1%. 99% should be the other allele. Or if you got 50-50, like we did with our fly example, about half of the normal wings, half of them are going to be the others should be involved. So it's panmitic mating means it's based on the allele frequency. Okay? All right, so what does Hardy-Weinberg tell us? Well, it's got this equation which says we can look at the genotypic frequencies here, and we can understand what the allelic frequencies were, or we can look at the allelic frequencies and move down. Okay, and so it says basically that if we think about gene pools at time 1, we can actually make predictions about what will happen at time 2. So it's a predictive model. We can say, okay, if this is going on, I predict that it should stay constant or that this should change. Or we can look at some time now and go backwards. We can try to figure out, we do this a lot in conservation biology, where we try to figure out what was the gene pool really like before humans came in and broke it all up or hunted it to near extinction or whatever we did. Same thing for diseases and those types of things. We can do all sorts of things where we can look, either take now and predict what it's going to be like. That's what geographic medicine does a lot of time with these viral or other types of bacterial types of invasions that we have, these episodic events, and they're predicting over time how big the population is going to be or how great that effect of that particular new disease is going to be over time. So they use basically forms of the Hardy-Weinberg equation. If you know the mutation rate, you know what mutants are going to cause the disease and which are non causational ones, then you can make nice predictions. All right. Now, given that we have this, there become two really starting points for any population genetics. Doesn't matter how fancy the techniques, whether it's just pure theory or whether there's actual data or not, but you have to know two things. We start with allele frequencies, and then we use the Hardy-Weinberg. And we take the Hardy-Weinberg, and the last 100 years has been modifying that Hardy-Weinberg by adding in selection, adding in drift, adding in mutation, playing with those, changing to having 4 alleles or whatever. Okay. So right now, what I want to do is look at the allele frequency, just a simple way to calculate it. There are numbers of ways to do it, but this is something you just should basically know, and it just gives you an idea what allele frequency means. All right, so let's say we go out into a population and we're able to detect 3 genotypes at a single locus, homozygous for the AA, big A, homozygous for the little a's, and then the hets. Okay, we're able to detect. We could be wrong, it could be whatever. We can somehow detect the hets as well as each homozygote. So we got 50 these, 20 of the hets, and 30 of the homozygotes. We're going to use what's known as the allele counting method, and that's literally what it is. We're going to count these up, and there's two big A's in a homozygote, right? So we count them twice. There's only one big A here, so we only count it once, and then we just divide by some other stuff, okay? So the allele frequency is literally 2 times the number of homozygotes that you're interested in, so either A, big A, or little a, okay? Plus the number of hets down here, okay? Everybody gets those. So we would only use one of these at a time, right? Because they're calculating the frequency of that, not both. And then just 2 times the total, okay? 2 times 100. Why is it the total? Because the total, obviously we want the total. 2 times that because we're diploids, right? So there's a chance that you've got 2 alleles, 2 big A's or 2 little A's or an A, and vice versa. So you just calculate it up. All right, so let's figure out what this is for this group right here. Let's just calculate it for big A. All right, well, we've got 50 of them, so 2 times 50 plus 20, okay, is 120 divided by 2 times the total. The total is 100 individuals, but there's 200 alleles. So 120 divided by 200 is 0.6, okay? So that's about 0.6. Now for little a, you could do the same thing. We'd put 2 times 30 plus 20, okay, right? So there'd be 80 here, and you get 0.4. Or you can obviously, if you've got 2 things, they add is just like Hardy did, P plus Q equals 1, and P equals 1 minus Q, or Q equals 1 minus P. I mean, it's simple algebra. So we don't actually have to recalculate all this up. We could just do a simple subtraction and get it, okay? Now what does this tell us? It tells us that big A ought to be involved about 60% of the matings, one way or the other. These should be involved in about 40%. That's what panmitic means. It means if you reached into this population and just closed your eyes and grabbed an individual, okay, you'd have about a 60% chance you'd have it least one big A and a 40% chance it would have a little a. That's really what it means. Allele counting method. Play with it a little bit. Simple. There's lots of other methods to use, but these are the simplest ones, and they get the whole point across. Okay. All right. All right. The next lecture set, what we're going to do is talk about the Hardy-Weinberg and how we could use it, and do give a couple of examples of actually using the Hardy-Weinberg itself to tell us interesting things about a population.